In light reactions, P2 uses light to extract electrons from water molecules, which when removed release oxygen, the air we breathe, and hydrogen ions, used to power ATP synthesis. The electrons removed from water, after being transported down a chain of electron-carrying proteins, pump hydrogen ions across the membrane. They are also used to help carrier molecules known as NADPH to deliver them to the enzymes that build sugar from water and CO2. P1 uses its harvested light energy for making NADP plus from high-energy molecules and ATP, which are later used in the Calvin cycle. Raising or lowering the pH from 8, the optimum pH at which photosynthesis occurs, will negatively influence the rate of the photosynthesis because Rubisco will begin to work more slowly. If the pH becomes too high or too low, such as 6 or 10, Rubisco will stop working altogether.
The first stage of the Calvin cycle, light-independent reactions are initiated. CO2 is fixated from an inorganic to an organic molecule. The second stage, ATP and NADPH, are used to reduce 3-PGA into G3P. Then ATP and NADPH are converted to ADP and NADP+. The reactions of a Calvin cycle add carbon to a simple 5-carbon molecule called RUB, or just Rubisco. These reactions use chemical energy from NADPH and ATP that were produced in the light reaction. Now, the final product of a Calvin cycle is going to be glucose. In the Calvin cycle, carbon atoms from CO2 are fixed and is used to build 3-carbon sugar. Now, this process is fueled by and dependent on ATP and NADPH from light reactions.